Alex mentioned some technical stuff and technicalities. I don't think my talk will be overly technical. The idea is to give an overview of what adaptive designs are and what they can do, what they can achieve, what they can't achieve, and what to think about when designing a trial with an adaptive design. So if you were expecting talk about technicalities, then this will be a major letdown, but I don't think anyone will have come to this and expected primarily statistical talk. Okay, so this is a lot of this is based on work that I've been doing for the last few years with excellent colleagues from the Trial Methodology Research Partnership Adaptive Design Working Group, and you will notice the odd shameless plug for some of our recent or ongoing work or papers throughout the slides. So Alex has already mentioned it's a joint webinar from the Health and Care Research Wales faculty and Treialon Cymru, which is this new initiative led out of the Centre for Trials Research. And the logo, which I have to admit I've seen for the first time when I tried to Google it for this, to include it in this slide, and all that Google came up with was Triathlon Cymru. Did you mean Triathlon Cymru? So I think Treialon Cymru needs a bit more publicising. Right. Shall we talk about adaptive designs? So this question comes up, or should come up, whenever you design a clinical trial. And the question is, should you make it adaptive or not? Well, being adaptive is generally a useful thing, not just if you're a chameleon. They're particularly good at it, although this particular chameleon doesn't seem to be very good at it. But it's also a useful thing for clinical trials. So the traditional way of doing trials, which I'll call the fixed sample design, is to spend loads of money, loads of resources on the design of the trial. Then you recruit participants for months, sometimes for years. And at the end of the day, you analyze, and so many things, you analyze the data, and so many things might have happened in between. And you were essentially not responding to anything, at least in theory, that has happened between the design and analysis stage. But that's not the most efficient way of doing trials. In adaptive design, you have this additional kind of feedback loop in there. So during the conduct phase of the trial, you undertake one or more interim data reviews, and based on the outcomes of these data reviews, you make adaptations to the way the trial is being conducted. So just a slightly different representation, rather than just recruiting one sample of participants at the very end, you include at least one or several interim analyses midway through the trial so that you can respond to various signals or other things that will appear, will become more evident as data accumulate. We'll look at what this actually means in a minute. First of all, we should note that adaptive designs are a relatively new thing. If you look at this graph, I appreciate it's a bit small. You can see that it started in 1978, so there were hardly any adaptive designs. This is a graph of all registered trials worldwide and across multiple trial registries and databases. So until mid-90s, late 90s, early noughties, there were maybe 3, 4, 5 adaptive trials a year worldwide. And then all of a sudden, in the late noughties, early 2010s, started seeing more and more, 20 or so per year. It's still not very much, and the different colors are different types of adaptive design. So it was still a bit of a niche design. And then all of a sudden, this happened. And this is obviously something that happened in 2019, 2020. You can see here a list of just one type of adaptive design called MAMS designs. We'll come to that later on. And you can see the first one was started in 2000, then another one in 2006, another one in 2008, and so on and so forth. until 2019-20, all of a sudden 20 new MAMS trials were started in just 1 year. Why would that be? Well, obviously, COVID made it necessary to rethink the way we do trials, and adaptive designs, platform designs have become way more common ever since, and I don't think it's going to change anytime soon, even though COVID trials might no longer be the main focus for the years to come. So what exactly is an adaptive design? An adaptive design is defined as a trial design that offers one or more interim looks at the data that were pre-planned. So at the design stage, you include the option to look at the data, look at accumulating outcome data, and based on what you find, you modify aspects of an ongoing trial. For example, if you find that there's virtually no evidence of effectiveness of the trial intervention, or it's very unlikely that you will find any evidence at the end of the trial, then you stop. That would be one type of modification, but it's certainly not the only type. The key bit of this definition is, however, that whatever adaptive design you plan, it's only a true adaptive design if you preserve the validity and integrity of the trial. We'll talk about what that means later on. So just looking at data as you see fit during the trial is not an adaptive trial. That's just a bad way of doing trials. So this relatively generic definition includes things like group sequential designs, which many of you will be familiar with, I think, but also more sophisticated methods like Bayesian adaptive methods. So it's a very broad definition. However, it does not include what I termed fully flexible designs here, where you can just change basically anything, any aspect of the trial without having pre-specified that change. So an adaptive design is not just about adding flexibility to the design. It is much more about planning that level of flexibility that you're adding to the design. And it really annoys me when smart people who know a lot about adaptive designs say that an adaptive trial or an adaptive design allows you to change the design. No, it does not. It does not allow you to change the design, because the option to change aspects of the trial is part of the design. So you do not change the design. Any change you make is within what you have specified at the beginning, and that is part of your design. So you're planning to be flexible. What you're doing is essentially you're planning the road ahead at the outset of the trial, and you include forks in the road. So this would be an interim analysis where, depending on the outcome of the interim analysis, for example, whether there is very clear evidence of efficacy or not, or whether there's very clear evidence of lack of efficacy or even harm or not at an interim analysis. Based on that consideration, you make a decision to either go left, which could be to continue the trial without any changes, or you turn right and you either stop the trial early, or you increase the sample size, or you make some kind of change that you have pre-specified in your design. And this is the right way of doing adaptive designs. This is the wrong way. So any kind of spontaneous changes just because you feel like it, that's not the right way. Leaving the road is not the right way of doing an adaptive design. There should be a road. You should only travel where there is a road. If you don't do that, your trial will end up in a car crash and doesn't deserve to be called an adaptive trial. Let's look at a few examples of adaptive design. So the first one is probably the most common one, the group sequential design. Some people wouldn't even call it adaptive, but it meets all the criteria of our fairly generic definition. So that's any trial where you have one or more interim analyses at which you look at the evidence in favor and against the intervention arm. If there is overwhelming evidence in favor of the intervention, and you cross a pre-specified threshold for your p-value or your test statistic, then in this case you would be in the green zone. Here you would stop early and conclude efficacy, because there's no need to collect any more data to come to a convincing conclusion that this intervention is truly effective. The opposite is the red zone down here. So if it's very unlikely that you will be able to demonstrate efficacy at the end of the trial, and if that becomes clear at an interim analysis, then you should stop, because it's not good use of resources, and it's also probably unethical to continue. And if you're somewhere in between, you would continue and gather more information, that is more participants. An example of that that's currently ongoing in the Centre for Trials Research is the Pronto trial. So the Pronto trial is a group sequential design. It's a two-arm design looking at adults presenting to the ED with suspected sepsis. It's one of the largest trials currently in infections and intensive care in the UK. The intervention is to add a biomarker test, a procalcitonin test, to the usual risk stratification using news scoring, and then there are two co-primary endpoints. So we want to show that this addition of the new biomarker test leads to an improvement in effectiveness, meaning that there is less use of antibiotics. But at the same time, we also test for safety, meaning that we want to show that 28-day mortality does not increase, which we do with a non-inferiority test. We defined group sequential stopping rules. They're called O'Brien-Fleming rules. There are other types of group sequential designs. That's one of those technicalities that I'm not going into today. The idea is to ensure that you control your 5% alpha rate, even though you're doing repeated tests. So if you don't stop the trial at the interim analysis, you will continue to do a final analysis and then test again. So despite doing two tests where you test part of the sample twice, you will not inflate your overall alpha. You can stop early for effectiveness if you meet both endpoints. So if you show superior effectiveness and the safety endpoint hasn't got worse. But you could also show superiority if the safety endpoint got better. In these cases, you would stop early for effectiveness and say the new strategy with the procalcitonin test is better than the current standard of care, which is just the news 2 scoring. There was also initially a stopping rule for futility. So if you could futility for both endpoints, then you could stop the trial early. We ditched that for reasons that are unrelated to the design itself. Okay, that's one example of an adaptive design in action. Another type of adaptive design is the MAMS design, the multi-arm, multi-stage design, which can be considered an extension of the two-arm group sequential design. So instead of just having a control and an intervention arm, you have multiple intervention arms. In this example, we have 3 active intervention arms, and at an interim analysis, you would make an assessment as to whether they are better than or likely to be at least promising compared to control. And if they are not, you would drop them and just continue randomizing to the remaining arms. In this example, treatment 2 is dropped at an interim analysis, so the trial continues as a 3-arm trial rather than a 4-arm trial, which is, again, similar to the group sequential idea. You drop any arm that isn't promising at all, and therefore you don't expose participants to treatments that are not going to work and that are never going to end up in clinical practice. Another example of a very recently funded trial in the Centre for Trials Research. This is like Pronto, also HTA funded. It's going to be a trial in participants having undergone endovascular lower limb intervention for CLTI. There are 3 arms to begin with, a clopidogrel only arm, and then 2 combination treatment arms. The primary endpoint is event-free survival, so we want to see how long people survive after undergoing the revascularization procedure. And events are things like acute limb ischemia, amputation, heart attacks, ischemic strokes, and mortality. So in this case, there is suspicion that the clopidogrel only arm might not do very much on its own and can probably be dropped early from the study, which is why we've included a futility analysis. This is similar to a MAMS design. It's drop the loser design. So essentially, we suspect that one arm might be inferior to the others. And if that is the case, we would drop that arm at the interim analysis. So we perform comparisons versus that arm at the interim analysis. And if there's a very low probability of that arm being better than any of the others at the interim analysis, then we drop that arm early. Okay, more types of adaptive designs. One that is relatively straightforward is the sample size reassessment. So when you do a sample size calculation, you often make assumptions or just sometimes just wild guesses about things like variances and correlations and all sorts of parameters that you don't know very much about. You might have a good idea what the treatment effect might be, but what the variance or standard deviation or standard error might be, not so much. And the idea here is to do an interim analysis and then reassess those variables for which you have very little information, about which you have very little information at the design stage, and based on that, adjust your sample size, which ensures that the trial isn't underpowered at the end. There's also a type of adaptive design called adaptive randomization. Here you adjust not the overall sample size or the number of arms as you would do in sample size reassessment or a MAMS design, but instead you adjust the probability of being allocated to one arm or another. So in most trials, you would do a one-to-one allocation. So people would be equally likely of being randomized to one arm or the other. Or if you have multiple arms, you would have a 33% chance in three-arm trial, for example. But in an adaptive randomization trial, you would re-evaluate that probability during the trial, and arms that are more promising would get a higher probability of participants being assigned to them. This is considered an ethical benefit by some, because if, as a participant in the trial, you are more likely to receive something that is more likely to work better. It comes with a lot of practical difficulties, so it's a bit of a contentious design, but it's something that has been used and is also being used in platform trials quite recently. So unless you end up with very extreme allocation ratios, such as 10-to-1, which is obviously an indication that you should have stopped that arm that is so unlikely to show any benefit anyway, unless you have such extreme allocation ratios, it can be worthwhile thinking about adaptive randomization. Another type of adaptive design is the population enrichment. This is a very useful design when you suspect at the design stage that an intervention might work better for one or more subpopulations rather compared to others, but you don't have definitive evidence to not include them. So what you would do is you would recruit from a wider population, for example, all patients with a specific with a certain condition, and then at an interim analysis, you would assess whether they all benefit equally from the intervention or whether it only seems to work in one subgroup, for example, those with a certain biomarker, whereas it doesn't work as well or not at all, or even is even harmful in another subgroup that, for example, doesn't have that biomarker. And based on that, you would then narrow down or not narrow down your inclusion criteria. There are many more types of adaptive designs, and I don't have 3 hours or a whole day or 3 days to go through all of them in detail. So just to mention a few: adaptive dose escalation and dose ranging, so those early phase designs where you're trying to find the right dose that is safe, tolerable, but also efficacious. That's something that is very often done nowadays with adaptive designs because you have great uncertainty around that optimal dosing range. Biomarkers are often useful in adaptive designs, for example, for things like biomarker stratification, or, as I've just shown, population enrichment based on a biomarker. You can do some more niche things like adaptive hypothesis, where, for example, you start with a non-inferiority hypothesis, and once you show non-inferiority, you can then move on to show superiority. You can combine different adaptations in the same design, and that's sometimes a good idea. So you might have something that allows you to stop arms early, like a MAMS design, where you can drop arms early if they don't show any promise. But at the same time, you might also have a sample size reassessment in your design if there was major uncertainty about some design parameters. So combining some, that is a few, not as many as you possibly can, is sometimes a good idea. And as I mentioned before, Bayesian designs, which I will not go into in detail today, are also one type of statistical technique that lends itself very well to adaptive designs. This has been an overview of the classical adaptive designs. And just to briefly recap on some of the benefits and challenges, it gives you added flexibility, so you don't turn a blind eye to what's happening during your 2 or 3 or 4 years of recruitment, but you actually allow yourself as a trialist to make meaningful decisions as you probably also would in medical practice, rather than just waiting for the end of the trial. They are an efficient way of doing trials because they can lead, they don't always lead, but they can lead to shorter trials. If you stop early, they can lead to trials that require fewer patients to draw the same conclusions, and they can give you more accurate estimates. Think of the sample size reassessment design, where you might be able to increase the sample size, but then have enough power to actually show the effect you want to show. They are also, for these very reasons, they are considered ethical because they are better use of resources, in that they make it more likely that your trial is a success at the end. And they will also reduce the number, they tend to reduce the number of participants receiving inferior interventions, not just the adaptive randomization design, where you shift the allocation ratio towards the more promising arms, but also designs like multi-arm, multi-stage, where if you can drop an arm that's clearly not doing anything early on, then you're not exposing participants to something that's very clearly not doing anything. Flexibility can be a bit of a double-edged sword. As I've said before, you can't just add as much flexibility to your trial as possible. You can't just make any adaptations as you go along, because at the end of the day, what will you actually have proven with your trial? Something that was a major obstacle and is now becoming less and less of an issue is that adaptive designs are resource intensive, in that they take more time to design. You need the expertise, statistical expertise, and software, but software and all sorts of tools, educational tools, are being created. They are now available to wide audience, and they're not all just for specialists. And that's something that is becoming less and less of an issue as more and more people, not only methodologists, but also clinicians, become more familiar with these types of designs. And yes, they are complex. Some of these designs can be complex. That can be a bit of a downside, and it can also lead to difficulties with the interpretation of results, but we'll come to that later on and see how that can be avoided. So let's have a quick look at another class of designs that is related to adaptive designs, but isn't in strict sense always adaptive. And those are the master protocol designs. So the master protocol designs are trials where you have an overarching master protocol, but then you can have kind of sub-trials with different sub-protocols. So what does that mean in practice? For example, a basket design. A basket design is a trial where you include people with multiple conditions in the same trial, where you're trialing the same targeted intervention. An example would be a cancer trial where you include people with different types of cancer, in the sense that you include, I don't know, lung cancer, breast cancer, bladder cancer. So that's what I mean by types of cancer. That's probably not a medically accurate description. But these different cancers, even though they sit in different sites, they might have things in common on a molecular level, and therefore they might be able to be treated with the same drug or biological intervention. So therefore, it makes sense to have a basket trial where all people with a certain mutation in their tumor, for example, will be included regardless of whether that cancer is in their lung or in their wherever. And therefore, you essentially create multiple sub-trials because you will also look at them separately. So how well are the lung cancer patients doing? How well are the I don't know, head and neck cancer patients doing? But you have this overarching master protocol, and then you have different sub-trials within that. And the umbrella trial design is kind of similar, but the other way around. So it's kind of the inverse basket design, I would say. You have a single disease. So for example, you have just lung cancer, but not all lung cancers are the same, right? So the same drug will not work for all lung cancer. So based on the inclusion criterion, people with lung cancer of a certain stage, for example, you will then stratify them by something like a biomarker, and depending on the strata where their individual tumor falls into, you will then give them a targeted intervention that targets that specific subtype. Again, you have multiple sub-trials, but also you will also be answering an overarching question about that type of disease, in my example, lung cancer. And then you also have platform trials, which are similar to MAMS in that you have multiple arms and you can drop some of them, but there's more to that, and I'll come to that right now. So platform designs are a bit like a souped up version of MAMS. So the MAMS, the multi-arm, multi-stage, that was the design where you have a control arm and then multiple interventions, and then those who don't work well, you can stop them early. In a MAMS design, you can do more than that. So you don't just stop arms early for effectiveness or lack of effectiveness or safety reasons, but you can also add new interventions. So here, for example, interventions D and E, they have become available after the start of the trial. So rather than setting up a new trial, they were simply added into the trial. This, of course, has statistical implications on things like type 1 error rate control. And there are methodologies that can address these. It's not always straightforward, but it is certainly worth doing. It's not just that you can add new arms and terminate arms. You can also, if one of the initial intervention arms turns out to be more effective than the initial standard of care, you can also make that arm the new standard of care. So you're almost creating a perpetual trial. You're creating a platform. You're not just creating one platform, you're creating the whole train station, and you can just add and remove individual platforms. I know that's not how a real train station works, but this is where my analogy breaks down. You can also add kind of a second sub train station. That's what we've got below the dotted line here. So for example, if you add another type of stratification, like a biomarker subgroup stratification, then you can also have a subgroup. So you're creating this whole environment where more and more people become eligible to be part of a trial, even if it's just one sub aspect of the trial. So essentially, you're creating something that is more inclusive than any two-arm standard trial could ever be. And this is something that is enormously efficient. So this quote here, that's a statistician friend of mine was interviewed by New Scientist magazine about these platform designs. And he said, the way we currently carry out clinical trials is like we're building a new football stadium for every single game, and it doesn't make sense. It's inefficient. So what we want to do is build more platforms and baskets and umbrellas where you have the whole setup phase, the whole infrastructure, the whole approvals, everything, writing protocol. You do that only once, and then for additional arms, you can write sub-protocols that slot into the master protocol. You can get sub-approvals, but essentially the whole trial machinery just keeps going and going and going. So here's an example of a scenario where you do 5 individual trials, each with a sample size of 100 per arm. So for example, trial 1, you might find that something is better than waitlist control. Okay, well, fair enough. Trial 2, it's a fail. Trial 3 starts later. Yeah, well, works, but it's a different type of control. And so you have all these different individual kind of pockets of evidence that you're creating. And that's not really efficient. What you want is a platform. What you want is something where you maybe have a slightly larger control arm, But then you have all your interventions that are being compared against that, and also in a way against one another. So this will be much more efficient, not just because of the operational efficiency that you only have to set up one trial, but also it will reduce your sample size because you don't need all of these different control arms, and therefore it will also be more attractive to participants because they're overall less likely to end up in a control arm. Here's another example of a real trial that we're doing in the Center for Trials Research, and that's a basket design, as you can probably tell from the illustration here. So the DOCTIS trial is a EU Horizon 2020 funded trial of adults with active immune-mediated inflammatory diseases that don't respond to conventional therapy and who have a positive biomarker profile. So these are the entry criteria. But if you look at the 6 diseases that are being included in this trial, you will notice that whilst they're all immune-mediated inflammatory diseases, they're rather different. And in the traditional way of doing trials, you would probably do 6 different trials for them. No, we're doing just one trial. We're doing one trial because as part of the project, we're developing a combinatorial therapy. And I can't tell you what that is simply because the overall project is at a stage where they haven't, the clever people who are working on that haven't fully figured that out yet. But there will be one combinatorial therapy that will be trialed in patients with all of these diseases. Well, no individual patient will have all of these diseases. So all of these diseases will be included in the basket trial. The primary endpoint is the proportion of participants who achieve disease remission at 24 weeks. And the criteria for that will have to be disease specific because remission means different things for different conditions. And then we will have, within each of the baskets, we will be doing an adaptive 2-stage single-arm study. What that means is we enroll patients, and then we do an interim analysis at stage 1. And if the remission rate is below a statistically determined threshold, we say, okay, there's very clear lack of efficacy. There's no point in continuing. And we just stop that basket, meaning we just stop recruiting participants with that condition. If the remission rate at that stage is above the threshold, we continue. And then at the end of the final analysis, we make a decision as to whether the intervention is likely to be effective or not, or efficacious or not efficacious. So what we're doing along the way, what you can do in basket trials more generally, is pruning and pooling. By pruning, I mean we cut sub-trials, in this case, baskets or conditions for which the combinatorial therapy isn't showing good enough response or remission. And then at the end, those who are still in the trial at the very end, we will draw conclusions about each condition individually, but we can also use clever statistical ways of pooling them so as to make an overall assessment about how well this type of combinatorial therapy works in immune-mediated inflammatory diseases more broadly. So now I've hit you with a lot of different types of adaptive designs, and the rest of my time, I'll just look at some more practical issues and things that come up when thinking about adaptive designs of different types. So one question that tends to come up when you discuss adaptive designs with trialists, methodologists, clinicians is, but do these designs introduce bias? And the question is, well, not if you know how to design and analyze them correctly. So what people mean when they ask, do they introduce bias, is what if you stop early and it's just like a random high or a random low? Because in any trial, if you looked at the trajectory of the evidence, it fluctuates a lot early on, and then it stabilizes later on as your sample size increases. So this is what this figure is trying to illustrate, that you have a lot of variability. But the answer is no, it does not. It does not increase your chance of stopping at a random high or random low, because the stopping criteria, if the design is done well, the stopping the early stopping criterion will be designed in such a way that it is really, really hard to stop early unless there is very clear evidence. So the strength of evidence in favor or against one arm or another has to be greater the earlier your interim analysis. But still, there's still a risk of underestimation or overestimation if you don't analyze your trial correctly. So any trial that can stop early for efficacy tends to overestimate the treatment effect when it does actually stop early. That is because if you did lots and lots and lots of trials of the same kind, some of them, there will be variation between treatment effects between all these trials, and some will be the upper end, some will be in the lower end, and if you have a stopping rule, you will essentially cut off one end of the distribution. So there is a tendency to overestimate the treatment effect with a stopping rule for efficacy if it stops early, but on the other hand, underestimated when it doesn't stop early. And it's the other way around for futility. But again, that's just if you look at the raw treatment effect, you have to adjust it statistically, and we'll come to that on the next slide. So this is something, if people ask, do adaptations introduce bias? Yes, they do. If you just look at if you use naive statistical methods to analyze it, then it looks like you do introduce bias, but that's something you can do something about. So it's not a reason to not do adaptive designs. So that's what I just said. If you are likely to introduce biases if you use methods that are not fit for these types of designs. For example, you will over or underestimate your treatment effect if you use a naive estimate, a naive method for estimating. So that's what your friendly neighborhood statistician might call the maximum likelihood estimator. If you've ever come across that term, that's a naive estimator that will lead to over or underestimation. But there are unbiased or biased reduced estimators that protect against that. It's the same with confidence intervals. If you use a naive method plus minus 2 times the standard error, then there is a chance that they are too wide or too narrow or just mislocated, as in they might be, the point estimate might not be in the middle. But again, there are methods to correct for that, and you have to use them with adaptive designs. And statistician worth their salt will know how to do that. Hypothesis tests. As I've mentioned before, if you look at the data repeatedly at an interim analysis, maybe another interim analysis and the final analysis, then you will have analyzed parts of the sample multiple times, those that were recruited at the beginning. And that inflates your alpha. And again, you have to correct for it, and you can correct for it, and you should correct for it in an adaptive design. And there's
5 minutes,
also,
Phil. Sorry,
thanks.
just to interrupt you.
No, that's great. Thanks. There's also ways of doing simulation studies to look at all these operating characteristics. So advice is work with a statistician. They should know how to address these things. The other slight worry is the risk of operational bias. If you do an interim analysis, you obviously should not disclose the outcomes of that interim analysis to people actively recruiting participants or the participants themselves, because knowledge of interim results may alter the behavior of investigators and recruiters and participants. Sometimes it's not explicitly sharing the information, but it might be mere speculative knowledge. For example, if you know a trial has an interim analysis and it didn't stop, that means that the intervention must be doing at least something, maybe. So that's that kind of level of information. And if there is even just a subconscious alteration in the behavior of how people recruit or how they approach, how they're whom they're more likely to approach when recruiting participants, this might lead to a heterogeneity in the patient population. So it's really key to specify who will know what about the interim analysis at what point and when, and what you need to do to guard against these kinds of biases is specify all adaptations in advance, restrict access to interim results as much as possible. For example, only the Independent Data Monitoring Committee would have access to that information during the trial. And very importantly, describe every adaptation that has been made or has not been made but could have been made. Remember, not adaptive design can also not trigger an adaptation. Describe that very clearly in your report. And there is an extension to, so this is now where I'm banging the drum for some of our own publications. So we've developed a consort extension specifically for adaptive designs, which contains additional reporting items in addition to the basic consort guidance, specifically to include things like how did you minimize operational bias? What types of estimators did you use? Just very briefly on the cost of adaptive designs. Some people are also worried that adaptive trials cost more. That's not generally the case. If anything, they might save you money because you might or might not be able to stop the trial early. What it does, however, is it adds uncertainty at the design stage because you will not know whether certain adaptations will be made or not further down the line. And a few years ago, I had a similar slide and presentation that said most funding models are still incompatible with adaptive designs. And now I've modified it, and now I would say some funding models are still incompatible with adaptive designs. But funders are becoming more and more appreciative of those designs, and some like NIHR, for example, also Wellcome and MRC are actively encouraging the use of adaptive designs in their calls, and they will acknowledge that you might have to run a simulation study. You will need more funding for your statistician to do that during the setup phase, and that is certainly improving over time. And there's also guidance out there, for example, from the Costing Adaptive Trials project on how to navigate these conversations with funders. I'm going to skip that slide in the interest of time, just to say that an adaptive design, even though I may have now sounded like they're a good idea for every single study, they certainly are not, because in some trials it won't work as well, simply because, for example, you have a long-term outcome. So if you have a long-term outcome, then you can't adapt on that, because otherwise it will take you so much time to actually collect a meaningful number of outcomes for a meaningful number of participants that the trial is over anyway. So there's no efficiency gain in including an interim analysis. So to come to the end of this, what you want to do with adaptive designs when thinking about them and including them in your next trial is you want to embrace the flexibility that they offer, but you want to avoid excessive complexity. So you don't want to include as many adaptations as you possibly can. And so this is something I've stolen from a paper by Frank Bretz and colleagues, this illustration where they compare your box standard 2-arm trial to a pair of scissors. It's good at doing one thing and one thing only, whereas an adaptive design is a bit like a Swiss Army knife, and it's more versatile, more flexible, and that's essentially what you want in your trial. However, what you do not want is this kind of Swiss Army knife, which I have verified is a real thing. This is not Photoshopped. You can actually buy this thing on Amazon. I don't recommend it. I've included a few papers that I think would be very useful if you want to learn more about adaptive designs. We'll share the slides. The presentation is being recorded, so you will have these slides. Feel free to have a look. These are papers that are targeting non-specialist audiences. By that, I mean non-statisticians, but also statisticians with not so much experience of adaptive designs. There's also, and that's my last slide, there's also Panda is a tool, an online tool, panda.chef.ac.uk, which has been created very recently and which is an interactive learning toolkit for adaptive designs, which gives you a lot of information about all sorts of aspects of adaptive designs in much more detail than I've just done right now. So it contains videos, just lots of explanations, but also for statisticians, it contains example code that you can just run and do simulations. So it's a very comprehensive learning resource around all things adaptive designs. And this is my very last slide. So bottom line is, I would suggest embrace the opportunity of including adaptive designs in your trial portfolio, and make sure you do it the right way. Make sure you include forks in the road rather than just taking a turn and driving onto the grass or against the wall. So this is based on a quote from a very famous baseball player who you might have heard of, Yogi Berra, who said, "When you come to a fork in the road, take it." Thanks very much.